Hello everyone, this is Kunika Chandra from Shivaji College, Delhi University. So in this video, we will be talking about the part 2 of our lecture on peptides. So let's have a quick overview at what this lecture would hold. So this topic on peptides was divided into two parts. Part 1 was covered in the previous video. In this one, we will be talking about protecting groups that we use on an amino acid before the synthesis of a peptide chain, and then we will be moving on to how to prepare a peptide chain with two methods: Merrifield solid phase polypeptide synthesis and the DCC method. Starting with the protection in peptide synthesis. So we know that a peptide chain is made up of several amino acids. Now, in an amino acid, we have two functional groups, NH2 and COOH, and both are very reactive towards each other. So reactive that sometimes in the same amino acid molecule, they both would react each other to form a compound. Now, when this happens, we cannot synthesize a peptide. So, in order to stop their reactivity, we used protecting groups on either the N-terminal or the C-terminal or both of them. So according to this point, we know that to avoid the reaction of NH2 group and COH group in the same amino acid, we would use protecting groups. A protecting group would be applied on the N-terminal and the C-terminal so that the reaction between both these functional groups do not take place. Now, protecting groups are of different types. To protect the N-terminal, we have a specific set of groups. The first is the BOC group, also called the T-butyloxycarbonyl group, and we can also use the thalloyl group. And to protect the C-terminal, we use a simple ester group. So we'll start with the protection of N-terminal first. Let's see how it's done. The first group that we use is the T-butyloxycarbonyl group, or in short, the BOC group. Now, this part is the protecting group, and it reacts with the N-terminal in this manner and would get attached to the N-terminal, and it would be called as the BOC amino group. So we can see that it has disabled the high reactivity of the NH2 part of the amino acid. Now when we have protected the amino acid from the N-terminal side, we can add other amino acid molecules to make a peptide chain. Now when we have made our required polypeptide chain, it becomes essential to remove this protecting group. So this term is called as deprotecting the protecting Boc group. So what we do is we simply pass this Boc protected amino group through trifluoroacetic acid or HBr in acetic acid, and this protecting group would get reduced to these two compounds, and the peptide chain that we have made would remove itself in an intact form here. So now here I've only shown a single amino acid molecule and not a whole peptide chain for the ease of understanding. The next protecting group that we will be doing is the thalloyl group. So as I told in the previous slide, I will only be using a single molecule of amino acid to show clearly how the protecting group works and not an entire peptide chain. So this thalloyl group is also called as N-ethoxycarbonyl thalamide, and as similar to the BOC group, this group is also protecting the N-terminal by undergoing a condensation reaction with the amino acid molecule. And after that, we'll get a compound called thalloyl amino acid, and we can see how this protecting group is protecting the N-terminal. Now when we have made our peptide chain, we would want to remove it from the N-terminal. So for deprotection, we will simply pass it through hydrazine. So this compound would get formed along with a free amino acid. And that is how we use thalloyl group in protecting the N-terminal of an amino acid. Moving on to protecting the C-terminal. Now, if you want to protect the C-terminal, that is the COOH part of the amino acid, we can do it simply through an esterification reaction. So, ester groups like methyl, ethyl, allyl, or benzyl ester protects the carboxylic end of the amino acid. So, it's really simple. We just need to do an esterification reaction. Here we can see that we have an amino acid and we have reacted it with alcohol and HCl to produce an ester. Now, this C-terminal gets protected by this group, and this is actually how C-terminal is protected. Now, when we have formed our peptide chain, we would want to deprotect this. So we'll simply pass it through a base and we'll get our intact peptide chain again. So now when we have learned how to protect the N and C terminal before a peptide synthesis, let's start with how we actually synthesize a peptide. A peptide means a chain of amino acids. So the first method we will be doing is the DCC method. DCC is actually called dicyclohexyl carbodimide and it is a coupling agent. So let's see how it's done. We have two amino acids with us, and as I told earlier, the NH2 and the COOH groups are very reactive. So we only want this one and this one to react together and not the N and C terminal over here. So we'll immediately use protection over them and protect them so that they do not react in the reaction. Now we pass it through DCC, which is our coupling agent, and we'll get this peptide formed with protected BOC group on the N terminal and ester group on the C terminal as we had protected it. Now when our peptide is formed, this is a peptide that I've made from only two amino acids, so it would be called a dipeptide. This is the product that is formed. Now, water would get expelled out and what happens is we get an amide bond. Now, DCC has a special feature in which it would react an acid and amine to give us an amide bond. So this is the property of DCC. And when we have this protected peptide, we would want to deprotect it to get our original dipeptide. So we'd simply get it by passing it through HBr. So in the next slide, we'll take an example of two amino acids and make a dipeptide out of them. So in this example, we will have a thorough understanding at how a peptide chain is formed. For simplicity, I have taken a dipeptide glycyl alanine, which is made up of two amino acids, glycine and alanine. So let's begin. We have taken two amino acids and according to the DCC method, we'll pass it through DCC, which reacts the carboxylic group and the amino group to form an amide bond. But before that, we have to take in mind that we also have the NH2 and the COOH groups over here, which are very reactive, so we need to protect them. So according to our understanding on our previous slides, the N terminal is protected by the BOC group. So we'll protect it like this, and the C terminal is protected by the ester group. Now when both of them are protected, we will get a protected dipeptide. We can see over here that the N terminal is protected from the BOC group and the C terminal is protected from the ester group. So here we have our BOC and ester protected dipeptide. Now we have to deprotect these groups to get our original dipeptide. So we'll simply pass it through HBr in acetic acid and let's see the details of how this reaction takes place. So what HBr in acetic acid does is, first of all, it removes the BOC protected N terminal in this manner, and it also reacts with the ester group from the C terminal, forming a CH3Br, and this part would get out in the form of carbon dioxide. So what we are left with is this part, which is our dipeptide. So this part would get out like this, which is free of any protecting group, which is our dipeptide glycyl alanine, and this is how we synthesize a peptide through the DCC method. Starting with the last topic of our lecture on peptides, we would do another method of synthesizing peptides, which is the Merrifield solid phase polypeptide synthesis. So when we read this, it says that this method uses a solid support like beads of copolymer to synthesize peptides. Now, what are these beads of copolymer? They are actually an interlinked structure made of monomers of styrene to make a Merrifield resin in this case. Now, when we are making a peptide chain with the help of amino acids, these amino acids would hold on to this Merrifield resin as a support system, and we'll keep on adding the desired amino acids we want in our peptide chain. And when our peptide chain is ready, we'll simply remove this Merrifield resin and get the peptide chain that we wanted to. So in the next slide, we'll see how it's done in detail. So in the Merrifield method of synthesizing a polypeptide, we would be doing it by 5 steps. Over here, I have taken the resin, and since this is a really bulky group, I would be using the word resin whenever I introduce this group in my reactions. So the first step stays protect N-terminal of amino acid. So first of all, we'll take an amino acid and protect the N-terminal. So the interesting thing in Merrifield synthesis is that we only need to protect the N-terminal of an amino acid because we will be doing our reaction from the C-terminal. So once we have protected the N-terminal with the Boc group, we would be adding the resin over here, and the next step says that attach it with copolymer through ester bond. So this amino acid gets attached to the resin with the help of an ester bond that we can see over here. So once we have this Boc protected N-terminal and resin is attached to the C-terminal, we will wash this copolymer to remove excess reagent. Now we want to add another molecule of amino acid to make a peptide chain. So what we'll do is, for that, we would remove the Boc group with our reaction with CF3COOH so that NH2 group gets free again, so that we can add another molecule of amino acid over here. This next step, which is the step 3, is add another BOC protected amino acid to the copolymer. So when we had this D-protected amino acid, like it's free to react, we would react it with another Boc protected. Now we have to protect it with the Boc group so that NH2 and COOH in the same molecule do not react together. Now we can see that the NH2 is free to react with the C-terminal of the Boc protected second amino acid we want to include in our peptide. So we will use a DCC, and as we had learned in the DCC method, it actually combines NH2 and COOH to form an amide bond over here. This is the special feature of the DCC method that we did. So we can see over here that now our peptide chain has 2 amino acids attached to it. So we have to make a tripeptide. So for that, we would be repeating our step 2 and step 3 again. So we'll have a quick recap at repeating the step 2 and step 3 because that is what step 4 is. So in step 2, we had a Boc protected amino acid which we wanted to remove so that another Boc protected amino acid could react with it. So repeating the step 2 and 3, step 3 was to react the free amino group with the Boc protected second amino acid. So in this way we can see that the third amino acid got incorporated in our chain, making a tripeptide that we wanted to make. In the last step, which is the step 5, we synthesize our required polypeptide chain. Now, as I told in earlier slides, when we make our peptide, we would want to remove the protection group and the resin also in this case. So all these tasks are done by a single reagent called HF. So as we can see, it has many uses. First of all, it cleaves the ester bond, and thus removing the resin from the C-terminal of our peptide chain. Second, it leaves the peptide chain intact, like it would not disrupt or break the bonds of our newly formed peptide chain. It would keep it intact. And third, it removes the POC group, that is the protecting group applied on the N-terminal, with ease. So this is how we use Merrifield solid phase polypeptide synthesis to make our peptide chain. We can repeat these steps to make any length of peptide chain that we want. So that was all in the part 2 of our lecture on peptides. Let's have a quick recap at what all we did. So first we started with protecting groups that we use to protect the N and C terminal of an amino acid because they are so reactive that they can actually react together and disrupt the formation of a peptide. So to protect the N terminal, we used two groups, BOC group and the thalloyl group, and to protect the C terminal, we used a simple ester group. So now when we learned how to protect the terminals, we could actually synthesize the peptide. The first method we did was from the DCC method, which is a coupling agent, and the second was the Merrifield method, where we were using a solid support of beads of copolymer to synthesize the peptide. So I hope that the topics in today's lectures were clear, and thank you for watching.